And I think with that, I will do an introduction. So Dr. Caballero received her bachelor's degree in biology at the Faculty of Science of the National Autonomous University of Mexico and completed her master's degree in micropaleontology and marine paleoceanography at the same institution, the same university in the Institution of Marine Sciences and Limnology. Her PhD in the Institute of Geography at the same university was focused on paleoenvironmental dynamics of vegetation from central Mexico. And this was followed by a postdoc with the Smithsonian Tropical Research Institute, which took her to Panama for this work, where she focused on statistical analysis of paleoecological data, and she was the team lead for the palynology imaging project there. And that was essentially how I came to meet Dayenari. It was about a year ago at a global bioimaging facilities manager training course in Cuernavaca, Mexico, where she gave this fascinating presentation on the work that she was doing in her postdoc with the Smithsonian. And she's now currently working as an independent data scientist for different institutes in different fields of environmental science, and I think is looking to transition fairly soon into in an industry position. So without further ado, I will hand it over, and I'm excited to hear the presentation. Thank you, Dayenari.
Well, thank you, Nikki, for the presentation. And well, thank you all for being here and letting me be here with you today. So I'm going to share my screen to start with the presentation. So is it okay? Okay. So today I want to talk to you about my participation in this big project that was taken at the Smithsonian Tropical Research Institute. So this project is about the study of palynology as an paleoenvironmental tool to reconstruct the past environments in different parts of the world. So I want to talk a little about the past, present, and future of this discipline. So to start, I want to talk a little about what is palynology. So I'm going to talk about mostly pollen, but I want to let you know that palynology also includes not only these tiny structures from plants, but also some algae and some fungal structures and even some marine organisms such as dinoflagellates and foraminifera. So our focus is going to be on pollen samples and pollen and spores, which are indicators of plants. And I want to tell you about well, this pollen is a great tool to study vegetation. Some has referred to the pollen as the fourth dimension of vegetation because they are present in the fossil records. So through the fossil record, we can access to different moments in time and to assign a certain vegetation type to a certain moment in time. So, well, let me tell you a little about what pollen is. Pollen are these structures which contain genetic material. So its principal purpose is to take the genetic material from one flower to another or to one plant to another. And because this process is often implies to travel, the plant needs to ensure that the genetic material is not damaged through this travel. So there is this coat that is covering the genetic material, that it's made of a natural polymer made called sporopollenina, sporopollen, and that it's very resistant to desiccation and degradation, and it's so effective that it can be preserved in sediments for thousands and even millions of years. So because of this particular conservation, we can access to the sediments and be able to assign this pollen to its parental plant. So let's talk a little more about this process. So different vegetation will produce different assemblages of pollen. So by tracking these changes in the pollen, we need to calibrate first the modern signal of pollen in modern sediments. So, the variety of pollen samples is enormous, and it's even bigger through time. So these are a few examples of how pollen might look like in the microscope. So as I told you before, this coat is very resistant. So when we find these structures, these sediments, we need to process first by throwing strong acids and basic compounds to disintegrate all the organic matter and all the other sedimentological particles. And as this sporopollenin is resistant to the acids, we can keep these elements and separate them for its identification. So in the natural world, as the vegetation changes in certain ecosystem or environments, the palynological signal will also change, and this will be accumulating in the sediments. So not every particle of pollen will reach its reproductive purpose, but a great amount of the pollen will remain on the sediments. So it will be staying in the soil, and then will be washed to sedimentary basins. And this is where we can recover this material. So we often use these cores to extract these tubular structures, which have different sedimentological features. So we usually, what we do is to take slices along these cores, and we analyze the content on each slice of sediment. So by tracking the changes in composition and in species and proportion of species, we can make a history of what happened around these basins. For example, and in this is one of the first environmental reconstructions made, which was using the different palynomorphs to characterize different physiognomy of vegetation. So we can track the transition between I don't know, something like a desert to some a savanna and into the forest through time. And we do that by taking into account different properties of the assemblage. So we are not only looking the species that are present, but we also make statistical approaches using, for example, the pollen influx, how much pollen is on each slide of the sediment, how many species do we have in proportion to each other. If this is a rich or a poor environment, we can know also if this was a stable environment or it was changing constantly, because we can measure also the velocity at which the ecosystem is changing. So these are only a few examples of how we can use these tools for looking into the past of our own planet. So that being said, well, this the first documented works of palynology as paleoenvironmental tool were made by Leonard von Post in 1916, I think. So more than a little more than 100 years ago. So this is not a new tool, but we will see how is it transforming. Now, the project that is working now at the Smithsonian within a lot of institutions, this is a big effort that is happening now. We have a project which is drilling 3 main points along the Amazonian River. And we expect to have on each point at least 1 kilometer of samples in the depth. So we expect to analyze at least 1 meter of one sample by each meter. So this is a lot of work that we have ahead. So the preparation started last year, and this is what this project is not going to be only about pollen. There are different indicators that are going to be analyzed in this project. For example, we have climate that way it's going to be approached by geochemistry. We also have tectonic, and we will have different indicators within the fossil samples. So I'm going to focus this talk in the pollen, which is the part that we were doing at the Smithsonian. So this is a photo of the first meter that was extracted from this project. This was the last year, I think in June. That was after a long period of preparation, we finally were able to extract this material. So as well, I was telling you before, this material needs to be processed before we start looking for the pollen identification. So this is how a fossil sample looks like in the fossil record. So we, after cleaning these samples, we put this material into small slides, so regular slides for looking at the microscope. And we're going to see that there's a lot of material in these samples. So we first need to locate which one of these corresponds to pollen or similar structures, and which one of these are just material, organic material that was the price of something else. So let me show you. This is how pollen looks like under a microscope. So this, as you can see, pollen has a three-dimensional structures. So sometimes it's not easy to identify only by looking at one single photography. To capture the entire ornamentation of each pollen grain. So in tropical ecosystems, the diversity of pollen is much more larger than we can find in forests in high latitudes. So for this purpose is that we need to ensure that we have a proper collection of pollen samples to have something to compare. So this is one of the first points that points towards the need to this pollen project. So this is, I think I didn't say that, but this project is called Pollen project because it's about palynology imaging, what we are doing. And so the idea is to have digital images of big enough collection of pollen to be able to recognize this in the Amazonian project that is taking place. So, well, as you can see, these sporopollen coats are empty because the acids will dissolve everything within and outside the walls. But the walls are very characteristic of each specimen. So, for example, in the left, we have this. This is a spore which is already extinct. We have no longer this type of spores in the present, and this we have this like striae along all the body of the spore. And in the left, on the right hand, we have Annona kerimola, which is the guanabana fruit. So you can see that the pattern is totally different. The wall has a pattern in the surface, but also you can see in the, I don't know if you can see the signal here. You can see in the walls, they have these little like columns that conforms the wall of the pollen grain. So that's those are also characteristic of each pollen grain. So as you can imagine, to identify these pollen grains is not an easy task. So I want to tell you a little about the history of how is this made. So I was telling you that the first pollen analysis as environmental tools were made by Leonard von Post. This is one of the earliest photos I could find, which is Tosti von Post. I think it's Leonard's wife. And she is looking through a microscope. You can see the lamp outside, which is separated from the microscope. And this is probably something similar to what she was looking at. Okay, so when we count the pollen, we see these particles in the sample, and we count how many of these elements we can count of each morphology. So, well, this was at the beginning of the last 100 years, and this is a photo of Carlos Jaramillo, which is the leader of this project, just a few years back. He's using, of course, more sophisticated equipment, but you can see in both photos that they have a piece of paper next to each one and a pencil where they are taking the same type of notes. Okay, so they keep counting in the same way from the last 1,000 of years. So we hope that this effort that we are making today is going to take the palynology to a whole new level. And by doing so, we are going to be able to analyze this information from different ways. Okay, so not only we want to have some reference of the pollen grains, but we also want to be able to have, I don't know, stop depending of these elements of these microscopes to be able to count in every computer. So, for example, for an expert, this process of counting pollen from a single slide will take maybe 8, maybe 12 hours for an expert eye, for a trained eye. So if we translate this to the work that we expect to have with the Amazonian project, this will be years of work, even having a team of people working at the same time. So the digital palynology will not only have all the material like into hand, closer to share and compare within palynologists, but we also expect to train an algorithm to be able to identify at least the most common pollen types in the fossil record. So that will save not only time, but it will be able to be consistent in how do we count pollen every time. And even if we are not able to identify all the pollen grains possible, if the algorithm can identify the half of the pollen grains, it will save already a lot of time, and we will be able to show in real time everything that we are not able to identify to another expert to share our experience and expertise in the field. So this is what we're pointing out, but we are still far away from this because we need to start by digitizing all the material that we have available. So let me show you. Well, this is another pollen type. This is one of my favorites. Actually, this is an acanthasia, the work from the General Justicia. And you can see that a single photo would not be able to capture all the ornamentation of this pollen grain. Okay, so we can appreciate that we have 1, 2, and there's gonna be another pore. This is 1, 2, and I can see. I think that there are only 2, but we have these like big pores, and we have ornamentation around the pore, and we have a different ornamentation in the peripheral area. So in the current way that we identify pollen, to be able to place this in a single photograph or a description is not very accurate. Okay, so we need to have this kind of images that are summing all this information, and this is actually a set of images that are taken at different depths using an automatic stage in the microscope. So every photograph is taken with a difference of a few microns, sorry, and well, no single photographs. It's illustrative of how the pollen looks like in the 3D structure. So we need all these photographs to characterize a single pollen grain. So this is the kind of photographies that the team is taking imaging in the Panamanian facility. And the challenge, the true challenge of this project is not how do we take these images, but it's in the volume of images that we need to take. So the Alan Graham collection is the biggest collection of tropical pollen grains, and it has more than 18,000 different species. So we need to digitize all these species to be able to train this what we expect to train this model for automatic recognition. So for each, well, let me tell you a little about this collection. Was started in 1954 by Alan Graham. So what he did it was to go to different herbaria and take samples of their flowers within which are already identified, and he took a sample of pollen of each sample, and he made this kind of slides for its microscopic analysis, and each slide was had this card with it, with all the information from the herbaria, the species, who did who took the sample, etc. So the first challenge was to have all this information in digital, and for that, every card was given to real people to read and fill these gaps in these specific fields in a database. So the same card was given to 2 different people, and if the information was the same by the 2 people, it was given by good, and that was like integrated to the database. And if the information was different, it was given to a third person, so to compare and to check by double crossing this information. In the cases that there was no consensus in the information, the card was given to the expert, and then we did it by hand. But it was the list of the cases. Okay, so once we had all this information, we crossed this information with the actual material in the Alan Graham collection, and what we came up with this protocol of how these photographs should be taken, and what will be the path of each slide to go, so it can be fulfilled all the process. So we have to take these images in 3 different techniques, which are bright field, differential interface contrast, and confocal. So we saw that that pollen had natural fluorescence, so we could take these images. We unfortunately find out that the fossil pollen did not preserve this fluorescence in all the samples. So we decided that pollen fossil will not be taken in confocal. But anyway, we continue with the process, and we find out that we needed to take more than 1 million images to complete our objectives, and that every image weighed in average about 1 gigabyte. So that was like the second big challenge of this project. because we had to take all this information through all a chain of processes. So we have a little cartoon of what we're doing in this plot. So we can see in cyan the collections that we are digitizing at the Smithsonian. All these like blue shade means that we are doing this physically at the Smithsonian Tropical Research Institute. We have 5 microscopes participating in this project. And as you can see, the first step is called Enrique, because Enrique Moreno is our first filter. He's the expert in tropical pollen. So he makes a first location of the pollen grains that should be digitized. So he makes this pre-selection of the material. He also is in charge of determining how well or bad preservation of the slide. If we should take another slide, or we can, or is the slide useful or not for digitization, or maybe is misidentified, etc. So once that he chooses the pollen grains, a team of palynologists is in charge of taking these slides into the proper techniques. And then we have this 2 workstation, because we have different brands of microscopes. We need to use different softwares. So we have 2 working stations, each one will specialize in one software, where we are able to evaluate the photograph and ensure that it is well taken. And we also crop the image, so because we can have 2 or 3 grains in the same scene in the microscope. So we crop, so we can dismiss this empty space, so it don't takes unnecessary space in the server. So once the images are okay, they can be preserved in the server of the Stripe, and from there they can be analyzed. We're going to be sending this information to Illinois, where there's a different team working with the automatic identification. So the first testing it's running on. And we expect to apply these models to the fossil samples that will be taken with the nanozoomer. Which are these fossil samples with a lot of different grains that are not identified. So another thing that I like from this project is that all this information will be available to the public through the Homero server. This is not only of interest for paleoecologists. There are going to be different fields, maybe the honey producers or the pollination ecologists that will be interested in we have access to this collection on pollen. And well, this is going to be free access. So well, I think that this project will benefit a lot of people around. Well, finally, well, no, almost finally, up to last Friday, that was the results that we have. So we have around 260,000 grains already digitized, with an average of 50 planes in depth. And which means that we have more than 15 million images separated. And we have already digitized at least 1 grain of around 6,000 species, which is almost 1/3 of what we were planning at the beginning. But we have also integrated new collections to this Alan Graham original planet. So we have at least 5 more collections, which are not that big, but they are summing new species to this project. And well, this is a photo of the palynologist team. This is a team that is rotating. So most of them has been just 1 year with us, and they left. but so it keeps changing, and we have people from different countries, mostly all the America, South America, and we're hoping that this team will keep growing with time. So we have, I think those are great numbers, and we hope that it's going to be even fastest in the future, because we have learned a lot through this project. And I also want to talk a little about how we grow in this project. So almost towards the end, I want to tell you a little about the bloopers of the project. So we've been working with different softwares, as I told you, and each software has different formats, and they keep different information from the image. So one of the problems that we have in the project is that we need to homogenize this information. And so, because we are going to analyze it all together, then we need to be able to put everything in the same language. So we found out that Fiji was a program which is a free source, and it's compatible with most of the imaging that we are working with. And well, actually, I want to tell you that we are looking for some expert in Fiji that can help us to deal with this information. Okay, so if you know somebody that would be interested in this project, tell him or her to contact me or Carlos Jaramillo. We will be happy to add it to the team. And well, I want to tell you about some other problems that we have faced, which, to be honest, the first time I talk about this, I didn't feel very well about, because I felt like I was telling all that can go wrong in the project. But I think it's important, because we struggled because we didn't expect this to happen, and we cannot expect this to happen because nobody talks about the problems. So I want to share a little of what we struggle with through this process. And one of the biggest problems that we have is that, well, the Smithsonian Institution, the Panama facility is the only facility of the Smithsonian Institution that is outside the United States. So this is a little bureaucratic, has a little bureaucratic implications, because when we first started to acquire the necessary equipment, we were like advised that it was better to do it from the central office in Washington. So Washington purchased the first confocal microscope, and this one in 2019. Maybe you remember something came up in 2020 that we weren't able to work for a while. And when we were finally ready to receive this equipment and to build the microscope in Panama, the central in the United States said, okay, but we cannot go to Panama because this is not our jurisdiction. So as the purchase was made in the United States, they didn't contemplate the trouble that sending this equipment to a different country would imply that this change of jurisdiction. So they told us that because we were in Panama, there was a central in Mexico that was in charge of the Mexico and Central America products. So we had to contact with Mexico. And the Mexico office tell that they cannot build or arm this microscope because they haven't made the selling. So, well, we eventually came up to an agreement. So we needed new equipment. So they agreed to build both microscopes. But well, and everything was okay. Okay, but this misunderstanding took for about 6 months to be resolved. So it was a little difficult to struggle with this. And also because we are not close to one of the main facilities for either of the microscopes, we always need to be ahead of what can happen. So as the workloading is big because we have to take so many photos I don't remember who made this law that if something can go wrong, it will go wrong. Remember? So that's what is happening to us. We have a little about it because a little of struggle because the use of the microscope itself, but also we have encountered new problems that the same owners of the microscope didn't were aware. For example, we were working with negative coordinates for the pollen grains. They didn't even know that they can work with negative numbers, but we did, and it was okay. So another problem that we have is that when something, when a piece of the microscope stops working, we need to wait for a little long. Meanwhile, the technician comes and try to fix it. If they need a new piece, for example, we need to talk to different people to bring them from different parts of the world. And for example, this is something that happened with the confocal. So the light bulb didn't work, but the lasers were okay. So instead of just waiting for the piece that needed to be replaced, we find a new way just to overcome this little problem. So I think that's everything I have. I want to thank all the people involved in this project, all the institutional and the people that it's been has taken us from the hands to be able to construct this magnificent project. And so if you have any doubt, you can write to me or write directly to Carlos Jaramillo. So thank you very much.
Thank you, Dayenari. That's amazing. I mean, I heard some of that already in Mexico last year, but I love hearing all about it. And I think it's important sharing those challenges. I think that it's very important that people realize that there are hiccups along the way. And if you can help them avoid those hiccups and those pitfalls, that's great, because there, the idea of negative data and not publishing negative data or not sharing negative information is wrong, because it's never plain sailing. There are always problems. So, if you can help somebody be prepared for some of those problems, it's a really, really great idea. And the cell phone fix for your lost light source is like, whoa, wonderful. So I will let's see if there are any questions in the chat first. Lots of thank yous, lovely. Yeah, plant stuff is really cool indeed. But yes, and I love the fact that this is focusing. So human health isn't just about, looking at human tissues and human cells. It's about the things that we eat. And, the pollen has a problem for many of us, particularly right now at this time of the year. For me, I have terrible allergies. So getting an understanding of pollen and what's been around in the past and how much more prevalent it is, it seems to be, I think, just we hear over here that pollen counts are going up just because of the whole global warming issue, is that the period at which plants can produce pollen is expanding. And then you hear about, the honeybee issues and colony collapse and the fact that they are being challenged as well. So something as tiny as a pollen grain has such a critical importance really in health just generally and well-being. So it's lovely to see a presentation that isn't focused on looking at a human cell or a mouse cell or something like that. So thank you.
I see Johanna has a question.
Go for it.
Yes, thank you for a fantastic presentation. It was an amazing project. It was so great to hear about it. I could just echo all the things that Nikki was just saying. Especially also thank you for sharing the experiences and the challenges you face. It's just so important to talk about these aspects. I wanted to ask a little bit about the data sharing aspect, what you're doing with this database that you're building at the end. I think that's hugely important and such a great output from this project. I was mostly wondering, is this the only project of its kind, or are there some sort of community standards of how you annotate the data or how these pollen grains are named or classified that you're referring to, or are you really setting the ground here of what these standards are for the community with this project?
Okay, we know of different projects that are working with automatic identification of pollen, but the difference is that they are working with small groups of species. So they have 20, sometimes 30 different species that they are trying to identify. So the difference is that we are working to this big scale. So we are taking a different modern and fossil species. And we are not competing. We are inviting them to join also to our project too.
And to follow up there, would then the idea be that this database you're building would then be also taking other people's pollen images, or is this database for the pictures that are coming from this project only?
We want to invite people to bring together all the imaging. So, for example, all these new collections that were added to the project, it's people that knew about our project and said, I have these samples too, can I take them? So we invite them to take their own photos and share the information. I imagine if that you have already taken the photos in a different facility, it will be also useful to add it to the collection. But I don't know if we have to see if the metadata is also in the same terms to be able to analyze it all together. But yeah, I think it's totally possible.
Yeah, thank you. That's really good to know that you're thinking about this in a sort of bigger scales as well. And because certainly the metadata annotations are a big challenge if you want to have the data be reusable in different contexts. Thank you. Good luck with the project.
I thought it was amazing to just see the slide, that original slide from the early 1900s that you guys are working from, many of these. It's just that must be incredible to think that somebody prepared that all those years ago, and in the whilst they did that, they put down the sort of what we now call metadata that is helping you all have a much better understanding of, you know, where to take
and
the
the
next step.
importance of documents, everything.
Yes,
because some of them weren't useful because we didn't know anymore where were they taken. So
it's, I mean, it's the constant challenge of how much is too much data, but how much is enough data. Because as you point out, exactly, as you point out, that the images themselves are humongous, and the metadata piece of it is a very small piece of it, but an extremely critical piece in order for it to actually have true value down the road, 100 years from now, 200 years from now, when all the rest of us have gone and we can't say what it was we did necessarily.
Yes, of course.
Great. Any other questions? I mean, I also like the fact that this is being done, you know, in a part of the world where it still remains relatively untouched. I noticed that some of the sponsors are drilling companies. Are their interests because they want to try and help preserve some of this history? Do you know? Do you have a sense of you know, where some of the funding is coming from and the motivation behind it?
Not really, because this is a huge project. For example, we at the Smithsonian, we have some donors which are particular that we show them the project and they decide if they want to participate or not. I imagine that different parts of the project has also different sources of funding. So I'm not aware of all of them. But yes, because it is about the Amazonian, it calls a lot of attention because it's the biggest lung in the planet. So if we can know, I think I forgot to say, but we are planning to reach the Miocene, which is about 23 million years ago. And we can track how it has been changing from the almost I don't know, at the different stages of the planet, with a hot planet, with a cold planet. So it would be very interesting to have an idea of how this big ecosystem responds to these changes.
Yeah, it's phenomenal. Awesome work. Great. Any other questions? Otherwise, I figure we'll let you have some time back, and yeah, we'll be in touch. I think we can help connect you with some folks around Fiji. And don't you, Vanessa? I think we've got connections, unless there are other people in the audience that want to share, but otherwise I think we can help connect you there.
Yes, that will be wonderful. Well, thank you for having me today and for the opportunity to share this project with your team.